which of these materials are fermented and which are cultured? Well, first of all, which one's cultured? The cheese. The other two, technically, wine might be considered a drink rather than a food, but for this exercise, we're considering it food. So the poor quality apple wine and the sauerkraut are fermented, not cultured. How do we know? By definition, a cultured food contains what?
Living organisms.
They contain live microbial culture at the time they are a finished product. So if they're being commercially produced, when they are finished, they contain live microbial culture. By the time you get around to eating them, they may not, because they go and sit on the grocer's shelf. The numbers of live organisms begin to decline, sometimes rather sharply. And as I mentioned last time, if you cook them, that's going to kill them. So you need to eat them raw. Now, we're going to be looking at different fermentation products that you produced last time. Remember what I said about each food product. You need to know the name of it. You need to know which organisms are responsible for the fermentation, and you need to know what the fermentation product is in each case. So this fermented food is called what? Sauerkraut. Yeah, you might call it other things, but it's sauerkraut. And what organisms are responsible for the fermentation process? For lab exam 1, where food microbiology will be incorporated, you have to know genus names. Species names are extra credit, and they're not on the name list. Only genus names are on the name list. So that class names, phylum names, kingdom names, and domain names. Anyway, what organisms? What two genera? So you need to know that. Leuconostoc, Mesenteroides, and Lactobacillus brevis, and Plantarum. Supposedly these organisms go through population changes over time. What we did was set up an enrichment. So we chopped up the cabbage very finely and put salt on it. The salt created a hypertonic environment, but then it caused the cabbage, the plant itself, the cells within the cabbage, to lose water due to osmosis. It's in the lab manual. Leuconostoc, Mesenteroides, Lactobacillus brevis, and Plantarum. Now, that's not to say those are the only things there, because we have done, when we streak the yogurt and buttermilk samples on BCT plates, but I have had students streak from the sauerkraut, from the juice of the sauerkraut, and they got lactococcus. So Lactococcus lactis can be in there, and there can be other kinds of organisms as well. So mostly Leuconostoc and Lactobacillus, and then in this case, where do you think they came from? Well, what did we do? You know, if you were absolutely sterile in your preparation, which would have been tricky, but you were chopping the cabbage very finely, and then all you did was gather that up and put it in the beaker. Forget what's on your hands for a moment, but you put it in the beaker with some salt. Where do you think the organisms come from? The cabbage. They come free with the cabbage. Now that's a deal, don't you think? What's very interesting is, yeah, there's bacteria on that cabbage. There are other types of organisms that live on the cabbage. And so you're going to have an opportunity, if you keep an eye on the containers that are on the back shelf, to see what's going on in those sauerkraut containers. At the end of our lab, I checked to see that you all had a nice brine formation. And because you were careful and chopped the cabbage very finely, you all have a lovely layer of brine. So that's the liquid that's up between the two beakers. This is an anaerobic environment. These guys are fermenting down here. They're probably producing some CO2, but they're mostly producing acid that's going to change the pH overall. As the pH drops, again, the population changes a little bit in terms of the organisms that are in there. But this is going to prevent the growth of fungi. So your sauerkraut containers are looking pretty good. I don't like to point fingers, but keep an eye on this one, because this one, as far as I can tell, has no brine. And you can already see a little bit of darkening on some of these cabbage pieces. There are a lot of fungi associated with cabbage because it grows in in the soil. There's a lot of fungi in soil. So if you don't do the enrichment properly, you don't catch it finely enough, get enough salt in there, there's not going to be fermentation happening. Instead, it's just going to be a big fungus mass. So we'll watch these and see what happens. Did I get it from up here? So, see what happens. Now, the other thing that you did with this was check the pH. So we had the pH strips. What was the pH to start? 6? Okay, so it was a little bit acidic even from the get-go. Sometimes it's closer to 7, but it's usually no higher than 6.5. So we don't actually start at 7, but we will check the pH again. It says here on day 6, this isn't, we haven't waited long enough. And so on Tuesday, we'll open these guys up and check the pH once more and see what happens. The pH is going to go down significantly, and the pH scale is a logarithmic scale, so those changes in numbers are quite significant. What is the fermentation product? Lactic acid. So these organisms that are associated with the sauerkraut production are predominantly homofermentative. They're going to produce lactic acid as their end product. Although it looks like they're also producing some gas in there. Now, what is the name of this material? Assuming that it works. This is our apple wine, or apple wine to be. And the organisms responsible for fermentation in our apple wine and also other types of wine and beer, champagne, etc. Saccharomyces cerevisiae. So now we have our apple wine container. One thing that we need to do is check in periodically to make sure there's enough water in the gas trap. So this one, the water level is quite low. We need to make sure that we increase that water level. Because we don't want air to get in. But we do want the material to bubble out. So if you can see the bubbles coming out of here, if I hold this flask and warm it up, the bubbles, what is that? CO2. These guys are fermenting. They're doing an alcohol fermentation, so they're producing CO2 and acetaldehyde, and then letting acetaldehyde be an electron acceptor, and from that they're producing ethanol. So ethanol is going to be trapped in the flask. CO2 is allowed to go out. Right now, these are really cloudy because the yeast are growing throughout, and so they're all over the place. Eventually, they have to turn to settle. And probably by next Tuesday, if you wanted to sample the wine, you can, and it's like sparkling cider. You can even put it on ice so that it's more palatable. Yeah, there's going to be some ethanol. Okay, so it'll taste pretty good on Tuesday, and then after that it kind of goes downhill because it begins to taste more and more like rubber stopper. And the yeast fall to the bottom and die there, and so then it begins to taste like dead yeast bodies. Not good. Okay, so CO2 goes out through the rubber tubing, and ethanol stays in the flask. If we were serious about this winemaking, at some point we would decant. So we would take the liquid off without disturbing the dead yeast bodies on the bottom, and have our wine product separated from both rubber stopper and dead yeast. We tend to forget to do that, and then it just sits back there and gets nastier and nastier. The same thing was happening here. Our fermentation, our organisms, so what organisms are doing the fermentation? We put in the same organisms. So again, Saccharomyces cerevisiae. They came in the little dried packet. They'd say Fleischmann's Rapid Rise. Anyway, yeast. So the fermentation products are also the same. So you've got to sample the root beer. Those of you that saw me open the bottle, a lot of effervescence, bubbles coming out, CO2. they're doing an alcohol fermentation, so there's gonna be ethanol in that bottle as well. Hopefully not enough that any of you went home tipsy. Anyway, the difference is here, we're sealing the bottle shut so the CO2 stays in. And that's what's gonna give the root beer effervescence as the carbon dioxide builds up in there. So then we also had our buttermilk mix. So again, this was about a quart of milk, maybe a cup of buttermilk, and 16 drops of rennet. It was a plant-based rennet, which is a proteolytic enzyme. And what it does is cause the milk protein casein to solidify. We were supposed to let it sit in a warm location, so I put it in a Ziploc bag and stuck it in the incubator. That's a pretty nice location, 37 degrees centigrade. Anyway, so what did it form? What's in the picture? Curds and whey. So, which is the curd and which is the whey? Yeah, the curd is the solid. You can remember that because we had curd on a cracker out there in the hallway. And then the whey was the liquid, which got thrown away. Sorry, but it did. This is talking about differences in cheeses. So there's information in the lab about the different types of cheeses, and you were supposed to kind of notice this as you were taking samples. Soft cheese versus soft cheeses differ from hard cheeses in that they contain more of what? Water. It's just water. And no, mice don't make the holes in Swiss cheese. And are these cheeses ripened or unripened?
Ripened.
How do you know?
I remember, yeah.
You remember seeing it?
I just remember you teaching, that's all.
Well, okay, but if we look at this, a couple of things. Ripened cheeses are those that are aged. They're allowed to sit around for a period of time, so the organisms present get to ferment more. And if you notice, this cheese has a kind of a rind on it that's a little bit discolored. This one also has some discoloration. And then here we see secondary flora or secondary microbiota. These are fungi that have been spiked in. Here you can see the spike marks. And here the fungus is on the outside. So they're aged. If they're ripened, they're aged at least. And then sometimes they have a secondary microbiota added to them, oftentimes a fungus. It may be just on the outside. It may be spiked into the center. If they're unripened, they're a finished product at the end of the initial processing steps. That was our curd on a cracker. It was large curd cottage cheese without the cream. So now we're looking at these plates. The bacteria genera that are most commonly used in the production of cheese, according to our cheese chart. Lactococcus, Lactococcus lactis. And then there are two subspecies. So we have Lactococcus lactis lactis and Lactococcus lactis tremoris. When that chart was first incorporated into the laboratory manual, all those Lactococcus organisms were classified as Streptococcus. But then with nucleotide sequencing, it was determined they were not related to Streptococcus, they were their own genus, and so now they are Lactococcus. And then, what fermentation product is causing the color change on the BCP lactose plate? Again, it's the fermentation product that gives cheese part of its flavor, the sour flavor. Lactic acid. So Lactococcus lactis is again homofermentative. They're producing lactic acid only, and so that's going to flavor the cheese. The other thing that we were supposed to do is compare our yogurt culture with our buttermilk culture. So this plate, it's hard to read which is on which side, but you have your own plates. So get your BCP lactose plate. Let's take a look at it. What we know from the lab manual, the yogurt organisms are supposed to be according to food microbiology textbooks, yogurt is supposed to contain Streptococcus thermophilus and Lactobacillus bulgaricus. But again, there have been classification changes, so it's no longer just Lactobacillus bulgaricus, it's Lactobacillus delbrueckii bulgaricus, so I stuck a D in there in the middle. I'm not sure why it's not usually incorporated into food microtext. But then I haven't looked at a food microtext recently, so maybe it has now. And then our buttermilk just says it contains live microbial culture, but it does not identify them. On the other hand, our buttermilk was used to make cheese. So we're going to assume that maybe the organisms present are the same organisms that are in most types of cheese, and that's Lactococcus lactis. Now, which side of your plate appears to have more lactic acid?
Butter.
You? How handy. We never know. The results that we get on this can be quite variable. However, Lactococcus lactis is strictly homofermentative. It's going to make lactic acid. And only lactic acid when it ferments the milk sugar lactose. So actually, it converts it to glucose and lactose first. Meanwhile, these guys, the yogurt organisms, can produce lactic acid, but they also produce acetaldehyde. Might be spelled right, might not. That looks fun, but anyway, acetaldehyde. An acetaldehyde is neutral. So presumably, if you have two different products, one is acid and one is neutral, then the amount of acid being produced should be less. Do you think those are the only factors that come into play, though? I mean, there was no guarantee you were getting exactly the same amount on the plate when you streak the plate. There's also no guarantee that you have the same amount of live organisms in the buttermilk versus in the yogurt. On the other hand, the yogurt was pretty fresh. It was just made the night before. I took the containers out of the yogurt maker and stuck them in the refrigerator. I found the buttermilk in the refrigerator here, so they should have been about the same temperature to start. So ideally, yes, you would get more acid from the buttermilk because Lactococcus lactis is strictly homofermentative, whereas the other guys are not. Now, I would like you to hold on to those BCP lactose plates. Hopefully you have space in your lab drawer so that they don't let them get away. They're worth 5 points when you turn them in, but I don't want you to turn them in today. I want you to turn them in next week because we're going to do gram stains for our physiological unknowns, and you get to do gram stains with these guys too. So you can find out what do the organisms look like that are living on that buttermilk plate. And if anybody would like to streak a sample from sauerkraut onto a BCP lactose plate, I think we still have some of that medium here. Maybe. Okay, so here we are again. What bacteria are the most common found in yogurt? Again, Streptococcus thermophilus and Lactobacillus delbrueckii bulgaricus. And the distinctive flavor, I mean, yogurt is sour, but yogurt has a kind of distinctive nut-like flavor, and that's the acetaldehyde. So acetaldehyde does give it a distinctive flavor. So does fruit if you add it, or vanilla extract, whatnot. Anyway, now there are some health benefits listed in the lab manual. I think I went over those earlier. They're on page 142. And then there are still nasty foodborne pathogen posters up. I talked about a few of the foodborne pathogens. There are a lot more listed there than I went over. So if you want to look at that and read about them, it's quite interesting. All right, so that's the food microbiology business. And again, we will have on our quiz Thursday next week, multicellular parasites and food microbiology. meanwhile, we're not quite finished with our food microbiology. So we're gonna do gram stains from the BCP lactose plates, and we'll turn those in next week. We will check the pH of the sauerkraut again next week, and you can try to scrape a little bit of material off the surface of the cabbage leaf and actually stain and see if you can see the organisms that are making the sauerkraut as well. It's a little bit hard to get them off of there. Okay, now we have 2 exercises that we have to do. What I'm going to do is explain these 2 exercises, and then I'll put the information on the board. We'll start with this one, which is determination of microbial numbers. So this kind of goes along with the lecture from last time, talking about microbial growth. We know that when microorganisms grow, we're talking about an increase in cell number rather than an increase in cell size. So in the picture on the left, you see very small cells. The picture on the right, you see the cells much bigger. Well, that's just because it's really easy with a computer to enlarge an image. It's the same image, you just made it bigger. Bacteria don't grow like that. Well, because it's important to know how many organisms are present, there have been a variety of techniques developed in order to determine how many organisms are present. Typically, we're talking about growing organisms in broth. So these methods have to do with looking at how many organisms, and it's always a matter of how many cells or colony-forming units per unit volume, and the unit volume is 1 milliliter. So one way that you can determine this is to do a direct microscopic count. This involves using slides that have grids etched into them, somewhat like your stage micrometers where you have etched. I'm not sure those are etched though, because they look like they're just painted, and it's coming off in places. They're kind of raggy looking. Anyway, you would have this grid, and you would count the number of cells visible in a set number of squares. And then you can use that to extrapolate and determine approximately how many cells are present in the entire volume of liquid, or how many cells per mil. You can't get a whole milliliter on the slide, so you're just going to be counting a small sample. Another way to determine how many cells are present, or to get an idea of how many cells are present, is to look at turbidity. Turbidity is the degree of cloudiness within the tube. And so there are instruments that can allow you to do this. They block the amount of light passing through. The turbidity will block the amount of light, and then the instrument will measure the extent to which the light is blocked. So they're looking at turbidity. Another way to determine the number of cells present is to actually pass them through a little aperture where you have an electronic signal as each cell goes through, and you get an exact count of how many cells are in there. So you get to count them one at a time. There are actually counters like this that count blood cells and separate them based on cell type. Pretty sophisticated. We don't do that here. But anyway, this is an image of a device called a Coulter counter, and it just counts cells as they go through a little opening. The problem with these methods, although they give you an idea of how many cells are present, they do not tell you how many cells are alive. So if you remember, when we talked about population count, there's a curve, so there's a lag phase, an exponential growth phase, a stationary phase, and an exponential death phase. The cells are dead, they're still going to be present, but usually if you want to know how many bacteria are present in a sample, you really want to know how many living cells are present in the sample, not just how many cells. So we're going to be using a method that allows us to determine how many living cells or viable cells are present in a sample. And the method we're using is called the quantitative plating method, or viable cell count. Be careful of that word viable. I've had students, I'm not sure whether it's because they don't know what viable means, and then when they're reading their notes later, they just randomly change letters. But I've had students tell me it's visible cell count. No, it isn't. You can't see cells. It's viable. And what does viable mean? Alive. Means living. So we're going to count the viable cells, the living cells. For this exercise, you're going to work in groups. The number of groups we have to use is dependent on how many bottles we have. So we have these bottles we have to use for the serial dilution setup. And if we only have bottles to do 9 groups, you guys are gonna have to join with other groups, okay? I doubt that we have 10 bottles, or 10 sets of bottles. Usually we only have 9. On the other hand, we have 10 cultures. We just can't, overlap the bottles. So the first step, if you're wanting to know how many viable cells are present, the first thing you're gonna have to do is dilute the culture. Part of the reason for this is because if you put a large amount of cell material on a plate, and realize this is also called the quantitative plating method, so it is going to involve putting samples on plates. If you just take your loop and put the bacteria on a plate and you just streak back and forth, what do you typically see? Think streak area 1. What do you see? But do you see colonies? No, you just see a solid mass of growth. The only way you're going to be able to get an idea of how many cells are present is to look at the number of colonies present, because a cell is the same thing as a colony-forming unit, usually, unless the cells form a set arrangement. At any rate, we're going to dilute, but we don't want to dilute the way you do when you streak plates. You dilute your culture when you streak plates, but you really don't know how much you're diluting, and it varies, because I've seen a lot of variations on theme relative to dilution. So we want to dilute to a known extent so that we can then determine how many cells we have to start with, how many viable cells. So we're going to generate what's called a serial dilution. Basically what this means is you're going to use a series of containers. You're going to transfer a known volume of liquid into the container, into each container, and each container has to have a specified volume when you start. So you actually will know how much have you diluted that sample. This is going to require some pretty careful measurement on your part. We're going to start with 3 bottles. Each group is going to have 3 bottles. You're going to be working in groups of mostly 3 people. A couple of groups are going to have to pick up these guys over here on the side, so there will be a couple of groups with 4 people, but that's okay. The bottles that you're going to pick up are supposed to contain 100 mils of liquid. However, what I can tell you about these, if you were measuring water in a graduated cylinder, and if you very carefully put water in the graduated cylinder to equal exactly 100 mils, you can read the meniscus. What happens when you pour that water into another container? Some stays in the graduated cylinder, and you can't help that. The other thing that happens is when these bottles go into the autoclave to be sterilized, the rubber stoppers have to be set at an angle. They can't be closed because they'll blow off. And so the bottles are open, and the chamber fills up with steam. So how much is in the bottles? Somewhere close to 100 mils. To make the math easier for us, we're going to fudge a little bit on the exact volume per bottle, depending on what we're putting into it. These bottles are going to be labeled 10 to the minus 3, 10 to the minus 5, 10 to the minus 7. So we're just going to set this up and get the exercise accomplished tonight, but you need to know how those numbers are arrived at. And so when we have our data to look at, I'll go through the process, talk about how the serial dilution is done, and how you calculate the degree of dilution at each step. You're also going to pick up 1 mil sterile pipettes. These are the pipettes in sleeves. Partly what I try to do in this lab is give you an opportunity to use as many different types of equipment as possible, not knowing where you might end up and what kind of equipment you might need to use at some future date. So it could be argued that this is rather old-fashioned. On the other hand, these pipettes are still being made, so they still work, and actually, I like them better in the sense that we know they're sterile. And they're going to be sterile throughout. If you're using a digital pipette, you're assuming that the tips you put on that thing are sterile, and in this laboratory they may be and they may not. But if you take a pipette out of a sleeve, the sleeve is sealed. These guys are sterile when you start. So we're going to do this exercise using this type of pipette. Although this is a 10 mil pipette, you're going to be using a 1 mil pipette. You're going to pick up 4 of these. Remember that you open the pipette at the end that has the cotton plug, and then be very careful that you don't let any part of the pipette touch a surface that you don't want to pick up contaminants from. So what you're going to do is initially transfer 0.1 mils of broth. So each group is going to be given a broth culture. You're going to transfer 0.1 mils of that broth into the water blank labeled 10 to the minus 3. To use the pipette bulbs, how many people have used these things? Did you use them successfully?
Yes.
Oh, good. That helps a lot. Okay. Because sometimes this is our biggest problem. Again, I can demonstrate this with a used 10 mil pipette. So these pipette bulbs have valves in them, and the valves are labeled. The one at the top is A for aspirate, the one down here is S for suction, and this one over here is E for empty. When you put a 1 mil pipette in, first of all, always put your pipette in the bottom hole down here. Don't ever stick your pipette in the A hole. Okay. When you're using a 1 mil pipette, it is possible to put the device into this bulb and bypass the E valve. You can push it all the way in so that it's right up against the bearing in the S valve. Then you can squeeze this E valve all day and it won't work. You're not going to get the thing to empty. So be real careful about that. The 1 mil pipettes have lines on them. They're either black lines or yellow lines. In the illustration or the picture up here, they're black. So you want to see these two black lines outside the pipette. Don't make those disappear. So you put that in, then you squeeze the valve at the top and squeeze the bulb, and you generate a negative pressure. Then this bulb is ready to suck, even though technically there's no such thing. It all has to do with the pressure outside, pushing up. It sucks, really it does. So then you put it into the volume and then very carefully squeeze this valve. When I'm using this pipette, this is a 10 mil pipette, so it's very easy to use because it's a big volume and the liquid moves very slowly. When you're using a 1 mil pipette and you squeeze the S valve, the liquid tends to jump in there very quickly. So you must use finesse. Very carefully squeeze the S valve. So what I'm going to recommend that you do, you're only going to pick up 1/10 of a mil. So what I'd like you to do, you put the pipette tip into your culture, fill the pipette to 0.8. And then when you get ready to transfer that into the water blank, you're going to empty it to 0.9. That means you're going to keep all the liquid down here at this end, which means you're going to have a very low probability of sucking bacteria into the bulb. Please don't. We don't want these bulbs full of broth culture. Okay? Now, after you've used the pipette, the first pipette, just slide it back into its sleeve, and then these are going to be tossed. This one not so much. This one is an agarose pipette, and it gets used over and over again. So then it tells you to close the bottle securely. Be careful not to push the rubber stopper in so far that you can't get it out. And you're supposed to shake it 25 times. You just want to thoroughly mix the cells with the water in the water blank. Then you're going to get a second sterile pipette, and this time you're going to transfer 1 mil. When you transfer 1 mil, you're filling the pipette almost all the way up. So again, please use finesse. Don't let the culture get sucked into the bulb. You're going to transfer 1 mil of liquid from the water blank labeled 10 to the minus 3 into the water blank labeled 10 to the minus 5. It says don't get the cotton plug wet. The cotton will slow the liquid down, but a broth culture will go right through that cotton. Try not to let it get that far. It's not supposed to. Then you close that bottle and shake to mix it. Then you get a third sterile pipette and aseptically transfer 1 mil, again, of liquid from the water blank labeled 10 to the minus 5 into the water blank labeled 10 to the minus 7. Now this pipette, this third pipette, if you keep it, you get to use it again. So you can carefully slide it back into its sleeve as long as you don't let it touch anything like your fingertips. Then close that bottle and mix it. So at that point, your serial dilution is done. You have a series of containers, known volumes transferred. Your serial dilution is ready to be used. Because this is a quantitative plating method, we're going to transfer samples from these water blanks onto agar plates. We're either using nutrient agar or tryptic soy agar, depending on what culture you have, and because we don't want to use up all of one type of medium. So it says here, in order to determine the number of viable cells, we have to transfer the samples from the water blanks to the surface where viable cells will be able to grow. And viable cells, again, are living cells. So this just shows the plates. Now, they're either nutrient agar or TSA. They're going to be labeled 10 to the minus 5, 10 to the minus 6, 10 to the minus 7. Notice the dilutions aren't the same, and they're not supposed to be. That is, they're not the same as the bottles. And then the first sample that you're going to transfer is coming from the water blank labeled 10 to the minus 5. You have a pipette that you used for that step, so you use that same pipette again, and you're going to take 1 ml of liquid from the water blank labeled 10 to the minus 5, and you put it onto the plate labeled 10 to the minus 5. If you're transferring 1 ml, then that isn't a dilution step, because you always determine the number of viable cells per ml. You transfer a whole ml, it's not a dilution step. But using that same pipette, you're going to go back to your 10 to the minus 5 water blank, and this time you're going to transfer 1/10 of a ml. And you're going to put it on the plate labeled 10 to the minus 6. It's 10 to the minus 6 because you're transferring 1/10 of a ml, and 1/10 of a ml is 10 to the minus 1. So that goes on the plate labeled 10 to the minus 6. Then you get a 4th sterile pipette. If you picked up 4 in the first place, you should have 4. And you're going to aseptically transfer 1 ml from the water blank labeled 10 to the minus 7 onto the plate labeled 10 to the minus 7. Once you have put your samples on the plate, you can use a glass spreader to spread them around. Actually, the ones that have a full ml, if you just very carefully tip the plate, you can get 1 ml of liquid to cover the entire agar surface. You don't have to use the spreader. But to get 1/10 of a ml to spread out, you have to use glass spreaders. Well, you might have to make glass spreaders because I don't see any back here. How many of you have ever made spreaders with Pasteur pipettes? Ooh, yellow cup.
I wish we were doing blue cheese.
So this is not a difficult thing to do, but don't everybody make a spreader because that uses up too much of our glass pipette. Each group only needs 1, hopefully. You can use it over and over again. So to make a spreader, first thing you're going to do is hold your pipette and put just the tip of it in the flame. You put it in the flame, it'll close the end of it, and it will cause it to melt. And it melts, and it falls over. Gravity does that. Now you have closed the pipette. You have a little piece of glass that's bending upward. The next thing you do is put it in the flame again, and just let it melt, and you do that. Now you have a spreader. Don't use this immediately because it's very hot. Also be careful. One of my friends recently said that he had a lab partner in chemistry, and the lab partner said, if there's one thing I learned in chemistry, it's that cold glass looks exactly like hot glass. Huh? It'll burn you good, so be careful with it. Anyway, once you have your little spreader, now you can use this to just spread. Okay? That's how they're used. If possible, you can use the spreader. You only need to use it for the plate that you put 1/10 of a ml on. So at the end of it, sterilize it, run it back through the flame. Be careful as you're sterilizing it. Don't hold it in the flame too long because it'll melt, and then you get all kind of weird shapes. But we can keep these, and then other people can use them later. Okay, so the Pasteur pipettes are in this box up here. Like I said, please don't everybody make a pipette spreader. Lots of fun, but don't go there. Okay, the idea is to get the liquid spread uniformly over the surface of every plate. Now you have 3 plates. The ones that have 1 full mil, you're supposed to tip your plates over because when you incubate, you're supposed to incubate auger side up. If you tip them slowly, most of the liquid will run to one side, so don't do that. Do a quick flip so that the liquid doesn't go to one side of the plate, and then you put them in the incubator. So you put them in a stack, invert with a quick flip, and then put them in the incubator auger side up. And then we'll do this business about reviewing serial dilution on Tuesday of next week when you have the results to look at. Okay, so that's our first exercise that we're going to do. The second exercise that we're going to do is the use of selective and differential media and replica plating. So, a little bit of background information. We had different media types that we were introduced to earlier, complex media versus defined media. So our complex media, as I said, they contain nutrients in crude form. Anytime you see extract of yeast, extract of beef, it doesn't matter. If it's an extract, you don't really know what's in it. You know there's some carbohydrate, some protein, some lipid, even some little bit of nucleic acid, but you don't know exactly how much. On the other hand, if you have a defined media have very specific ingredients and in specified quantities, so you know exactly what's in it, assuming your chemicals are pure. Some new categories that you're going to be introduced to tonight. The first one is selective media. Selective media are those that promote the growth of certain types of organisms while inhibiting the growth of others. If media are selective, they promote some growth and inhibit others. Have we used any selective media?
Yes.
Any of you that have written up your soil enrichment assignment know that yes, we did. We used selective media because we had Pseudomonas enrichment medium and Azotobacter enrichment medium. They were designed to promote the growth of certain organism types while inhibiting the growth of others. Tonight, during our replica plating exercise, you're going to be introduced to a whole series of different media that are selective. But that's not all they are. Differential media, this is another category. Differential media are those that allow multiple different types of organisms to grow, but cause them to look different. Usually it has to do with pH indicators. Sometimes not, but you're going to have different organisms. They're going to appear different because they're doing something different on the medium. Have we used any differential media? You might not know it, but yes, this is differential medium. Organisms that produce acid are turning the pH indicator yellow. If you put a fungus on this plate, the fungi don't produce acids, and the medium stays purple. We should have grown some brie. The brie organisms are very nice. The penicillium grow nicely on the BCP plates, and they just stay purple. So yes, we have used differential media. That one. Well, so the media types that we're going to be using for the replica plating exercise are selective and differential and complex. They all have extract of something in them, different types of things. So what does this tell us about media categories? They overlap. They're not exclusive of one another. Now, the other objective, so one of the things you're going to do is learn about some different types of media, how they work. Again, I could go through and explain this to you ahead of the exercise, but it's much more useful to explain it when you have data to look at, so that you can see how the media work. How are they selective and how are they differential? So we'll do that next week. The other thing we're doing is learning a technique called replica plating. I think there was a replica plating instance in that parasite film, but I'm not sure anybody was looking at it. Probably not. You were all doing morphological unknowns. Basically, what replica plating involves is setting up some kind of a device that you can use to pick up a pattern of colonies. So instead of picking up just one organism at a time, you pick up a whole pattern of colonies, and you transfer that pattern to a series of plates. We're going to be using velvet-covered blocks. They're supposed to be wood blocks, they're actually PVC drains, but they're covered with velvet. You have to set this thing up, and then you're gonna very carefully touch this to a set of colonies on a plate, and then transfer the pattern to a series of additional plates. You want to do it as aseptically as possible, but the real advantage to this is that you get to transfer 5 different organism types all at the same time onto a series of different media, instead of having to streak each organism type onto each of those different plates, which would have taken you 5 times 5. Did we do 5? I think 5 different plates, 5 different organisms. You're going to spend a lot of time streaking plates. Instead, you're just going to pick up the whole set and transfer. Boom, boom, boom. So it's quick, it's easy, saves time, it saves media. If you're working in a laboratory, clinical lab, or research situation, it's going to save money because it's really quick. For this exercise, you're going to be working in groups again. For this exercise, I made up 10 replica masters, so you guys can be a separate group if you want to. So we should have 8 groups of 3 down the middle. So each table is 2 groups of 3. There's a group of 3 on that side and a group of 2 on this side. Assuming our replica masters worked out well. So we're going to set up a series of differential media. We're using mannitol salt agar, McConkey's agar T7, and EMB. And then at the end we have a nutrient agar plate. Nutrient is not selective or differential, it's just a general medium. You're going to label these, and one of the things we want to indicate on these plates, not only what they are, but you're going to put a little mark at one edge to indicate the top. The replica masters are already marked. They just have a black line that indicates the top of the plate. You want to be careful about aseptic technique, so you're going to assemble your replica plating block. That means you have to put the velvet on the PVC drain without touching the surface. So you have to be careful to how you handle that velvet. We hold the velvet in place with a hose clamp. And the hose clamps are set to the right diameter, so they just slide on very easily. So you just be careful not to touch the velvet surface. By the way, put your velvet on the block, hairy side out, so that the little velvet hairs will pick up the bacteria. Now, that block, once it's assembled, is going to look like this, except that the velvet probably isn't white. Then again, we had red, we had blue. I never know what it's going to look like one semester to the next. But you can see how this is assembled. So the furry side of the velvet is out, the hose clamp is on. You want to slide the hose clamp down such that you have about this much space above that screw apparatus so that it will set into the plate. And then what you're going to do is hold the velvet up and press your replica master to the velvet surface. And you do this only one time. Keep track of where the top mark is. And you might think that it would be useful to put the replica master on the tabletop and press down on it. That's not helpful because you can't tell how much pressure you're putting on it. If you hold it this way so the velvet is exposed and press the plate against it, you can see that the velvet hairs are making contact with the colonies. Whatever you do, don't do this. Put it in there and smear it around. Okay, so then holding that replica block still velvet side up, but now with a colony pattern on it, you're going to bring each of your different media types and press it against that velvet so that you can see the contact between the medium and the velvet hairs. So, caution, don't twist or move the plate surface against the velvet because you'll wreck your colony pattern. Don't push it too hard because if you mash the agar, that will damage your colony pattern too. You just want to pick up enough cells on the velvet and then transfer those cells to each of the different media types. I'll put the order that you're going to put them on the board. What's most important is that the nutrient agar plate is last. So typically we do mannitol salt and then EMB, Churchitol 7, McConkey's, and then nutrient. But you only touch the master plate once. And again, keep track of where the top is on each one of these. Now, we're going to put these all in a stack and tape them together. including the master. The master stays with your set, and we're gonna put these in a cardboard box. The reason we have to do this is because the selective and differential media will give you the best results if you read them 24 hours after inoculation. That is, they're gonna go, they're meant to be used at incubator temperature, but you read them at 24 hours. Well, our lab is tonight. You don't come back in here until Tuesday. So I don't want those things in the incubator. That's too long. What we're gonna do is put them in a cardboard box, and I'm gonna hide them in the refrigerator, and then I'll come in on Monday evening, take them out of the refrigerator, and put them in the incubator. So they should be right when you guys look at them on Tuesday. So we're gonna put them in a box. This is the physiological unknown. As I said, I'll put this on the board. We're gonna have a set of tubes. We did this with the morphological unknown, same situation. Although the physiological unknown set is full of organisms that could be considered potential pathogens. So please be very careful how you handle these. Each tube that you pick up, instead of having one organism type, has 2 organism types. Your job is to separate them. So this is kind of like the mix A, mix B business. That means you must streak your plates properly. So what I'm going to ask you to do is pick up your tube. Each person is going to get a tube. Remember, carry them by the glass, not by the caps. You're going to take your tube to the workstation, put it in a secure location. You can use your wood block, or you can pull one of the white racks from the shelf at the back. They're up on top. Anyway, set them down and then get the medium that you're going to use. We're going to, for the most part, be inoculating 2 auger plates, and I'll put on the board which types of media you're going to use for this. We'll use at least one that's selective and differential, and one that's not. I think for a couple of the samples, you'll inoculate 3 plates. So this says everybody's going to inoculate 1 plate of either MacConkey's or T7, not both. Please don't use both, because we don't have enough media. This is a big class. There's 29 of you guys in here, so please be conservative with the media. Don't waste media. You make a choice. MacConkey's is pink, T7 is green. Don't use both. In addition to that, you're going to inoculate tryptic soy agar, but the MacConkey's in T7 and also the mannitol salt, again, those plates have to be read at 24 hours. So those plates from your physiological unknown also go in the box with the replica master and the stacks of replica plates, so that they're in there for the right amount of time. Some of you will be inoculating nutrient agar plates. Some of you will be inoculating Mueller-Hinton plates. Those can go directly into the incubator. Blood agar plates also can go directly into the incubator, so they don't have to go in the box. So the only colored plates that go in the box are mannitol salt, MacConkey's, and T7. Some are doing nutrients, some are doing Mueller-Hinton, some are doing brain heart, some are doing blood agar. Those can all go in the incubator tonight, and I'll take them out tomorrow. The other thing is, please put your physiological unknown culture tubes back in the rack. Don't keep them, don't hide them in your drawer. We need them back in case you need them again later. And basically, the completion of this little project requires that you identify 2 organism types, genus and specific epithet, and that you describe how you identified the 2 organism types. Part of that how is morphology, so you have to do a gram stain and an indirect. That's it. No endospore, no acid fast, no capsules. Just gram stain and indirect, that's it. You're going to record cell size, shape, and arrangement. It's a good idea to do a KOH along with your gram stain to double check to make sure that it's right. And you're going to describe all the different tests that you get to do to identify. There's a bunch of them, and it's all in the lab manual. Now, this is something we're going to be working on for the next 2 weeks. It's not going to happen really quickly, but don't